Now that's Huggie Pacino certified.
Well, hello y'all, this is Huggie Pacino with another video. Very interesting article here I read. So it's going to be a very long read, so I want y'all to just really get yourself together, have a seat, enjoy it. And make sure you share this because this is very important. How pieces of live human brain are helping scientists map nerve cells. An audacious project aims to figure out how humans are different from other creatures, okay? The golf ball sized chunk of brain is not cooperating. It's thicker than usual and bloodier. One side has a swath of tissue that looks to my untrained eye like bristles. Nick Deese, the neuroscientist charged with quickly cutting the chunk into neat pieces, confers with his colleagues. We can trim off that ugliness on the side, he says. The ugliness is the brain's connective tissue called white matter. Alright? To produce useful slices for experiments, the brain tissue must be trimmed, superglued to a lipstick size base that then fed into a lab version of a deli slicer. Alright? But this difficult chunk isn't cutting nicely. Dee and colleagues pull it off the base and trim it again and reglue. Alright? Now you're talking about playing God. This is it. Half an hour earlier, this piece of neural tissue was tucked inside a 40-year-old woman's head on her left side, just above the ear. Surgeons removed the tissue to reach a deeper part of her brain, thought to be causing severe seizures. Privacy rules prevent me from knowing much about her. I don't know her name, much less her first memory, first favorite meals, or sense of humor. But within this piece of tissue, what the patient generously donated and are clues to how our brain, how all of our brains really create the mind. Dee's team is working fast because this piece of brain is alive. Some of the cells can still behave as if they are part of a person's brain, which means they hold enormous potential for scientists who want to understand how we remember, plan, behave, and feel. After Dee and his team do their part, pieces of the woman's brain, scroll up here, will be whisked into the hands of eager scientists, where the cells will be photographed, zapped, and electricity received of their genetic material and even infected with viruses that make them glow green and red. It's all part of a project at the Seattle-based Allen Institute of Brain Science, funded largely by private money plus some U.S. government grants. See, why is it gonna be funded largely by private money plus? Come on, like for real, like just tell us who's behind it. We already know this got to be a bigger source behind this whole idea. Anyway, now in its 6th year, the project relies on a network of scientists, neurosurgeons, and patients who are willing to donate brain tissue removed during surgery. The ultimate aim is to answer one of the biggest questions in neuroscience: what makes us human? Alright? The answer won't be simple, but already the project has turned up hints in what makes the human brain so powerful. Live tissue experiments have revealed cellular quirks that may be specific to primates and have turned up in new details about mysterious types of nerve cells or neurons. Other tantalizing discoveries show that humans and mice have very similar number of neuron types. That's crazy. This kind of detailed cellular reckoning is the necessary early step on path to understanding human thoughts, behaviors, and abilities. We want a complete description of all the types of neurons, says Christoph? That's a weird name, Christoph. I didn't know there was I know it was a way to this Christopher. I didn't know it was a Christoph. That's crazy. Christoph Koch. Chief scientist and president of the Allen Institute of Brain Science. Steady progress over the last 6 years show that answers are within reach. Once order is given to the tangle of neurons that populate our brain, scientists could turn their sights to a bigger mystery, like how those cells create our memories, emotions, and even consciousness itself. Rush hour. On the morning of May 14th, I waited outside the basement operating room at the University of Washington's Harborview Medical Center. Inside, a neurosurgeon was cutting deep into a woman's brain. At 10:15, the wide swinging doors and the doctor carried out a clear plastic jar with an orange cap. Settled at the bottom of the liquid, inside what was a bit of brain, gently sloshing around with the motion. Tissue procurement team member Tamara Casper was ready with a cart that carried a blue cooler, the same kind I have in my garage. Wow. On top and 2 gas containers below. The piece of brain has tinged the clear solution pink. Look at that. That's crazy. Can we get a good look at this? Can we zoom in on this? I can't zoom in on this. I want to zoom on this bad so we can get a good look at this. That's the best I can get. A chill jar left holds a sample of brain tissue minutes after it was removed during May 14th surgery for epilepsy in Seattle. At a lab across town, the sample was trimmed and cut on ice, middle. Wow, that's crazy. Once trimmed, it is glued into a holder the size of a lipstick tool for more slicing right. That's crazy. Wow, unbelievable. It was a colorful reminder that this tissue had minutes earlier been inside a skull, where it was helping create a woman's mind. The scientists have other methods to mimic human brains. Brain organoids, small balls of neural tissue that are grown from stem cells, right? And an animal raised in the labs have been immensely helpful to neuroscientists. There are real values here, real value there, says Allen Institute neurobiologist Ed Lean. But what they're not good for is studying the specifics of final product in a matured brain. This particular sample submerged in the pink liquid had spent 41 years plotting a woman's life. It's hard to emphasize how different this is, Lean says of the project. Other laboratories have studied live tissue removed from the human's brain, but none have scaled and systematized the process as much as this group in Seattle. To me, it's almost mind-blowing that we can study the human brain outside the human brain, says Ryder Glenn, a neurosurgeon at Swedish Medical Center in Seattle who collaborates with Allen Institute scientists. Gwen treats people with epilepsy. Medication doesn't always stop his patient's seizures. In severe cases, surgery can be a patient's best bet. In some of these operations, the surgeon cuts away healthy brain tissue to help the spot deeper in the brain where seizures first start. Surgeons peel away the skin and remove a cookie-shaped piece of the skull, exposing the temporal lobe of the brain, and stretch the outermost layer called the cortex. Often a large piece of the temporal lobe comes out, Gwen says. Some of the neural tissue goes to pathologists. The rest is typically tossed as a medical waste, unless Allen Institute scientists can get their hands on it. Alright? The tissue is terribly scarce, Scott says. Early on, colleagues, including many Allen Institute researchers, were skeptical that enough samples could be found and brought to the lab in good shape. But after 140 surgeries, more than 30 this year alone, it's clear that these brain samples survived the journey beautifully. Wow. As soon as the sample came out of the ore, Casper hooked up into the oxygen and carbon dioxide gas to keep this tissue alive in a liquid. An artificial spinal fluid. I never heard of that before. Then she was off pushing the cart through the hospital with one hand and texting the Allen Institute team with the other. The car was loaded with a white van modified to safely hold combustible gas containers. And with that, the bubbling brain bit was on its way. The van threaded through heavy rain-soaked Seattle traffic back to the lab where Dee was ready, scalpel in hand. All right, so they basically got it together. And if you always notice, these people that be doing this research they have like foreign names and shit, but that's just another story. I ain't gonna get into that. Alright. Use the suction tool to get a good seal on a single live neuron's brain so you can see how the cell behaves. That's crazy. Alright. Crosstalk. After that frustrated start with the uncooperative piece of brain, Dee finally gets enough slices for multiple experiments. A 1 hour rest helps the cells recover from the trauma of being separate from the brain. The slices go up to the second floor lab where some pieces are placed under a powerful microscope and prodded with electricity to study how these live human cells behave. The researchers hope the behavior mimics what the cells did while they were inside their former on the skull. 6 scientists sit at a rigs. Each one of the microscopes mounted inside a black three-sided box. In each rig, a researcher hunts through the woman's brain tissues for healthy cells, nice and plump, with just the right amount of visibility against the background tissue. As you can see here, they got a chart here. Scientists can observe neighboring neurons. Left shows up to 8 pipettes. Is that how you say it? Pipettes, I'm assuming? Each touching a cell's nucleus to reveal complex cellular conversations on the right. Alright. Once they find a good one, the researchers try to latch on with an impossibly thin tube of glass called a patch clamp. The technique forces a cellular conversation, which is carried out with electrical signals that move between cells. To get the conversation going requires injecting an electric current into the cell, wow, and then measuring how the cell responds to the artificial message. All right. Most of these rigs measure the reactions of one neuron at a time, but in the back part of the lab, researchers Lisa Kim pilots a futuristic setup of glistening metals, tangles of blue and black wires, and 8 needle mounts, all point at a different part of the brain slice. While I'm there, this mega rig is eavesdropping on a kind of party line between 7 neurons. Kim is zipping electricity into each one in turn to see how the signals transmit to its neural neighbors. All right, this is unbelievable work. Using a multi-station rig at the Allen Institute, that researchers can observe a group chat among a handful of live neurons in response to an injected electric current. Unbelievable shit. The electrical zings of these neurons often clues about their identity and their relationship. One of the 7 cells respond when a neighboring cells gets an electrical zap, a hint that those cells communicated while inside the woman's head. Other clues come from the information about the neurons and elaborate gangly shapes created by the signal-sending axons and receiving dendrites. Each neuron reminds me of an impossibly complex map of rival river tributaries. All right? An even stronger sense of cells functioning come at the end of the patch clamp experiments. Working the thin glass tube again, a researcher can suck out the nucleus of each live cell. The theft kills the cell but obtains a record of which genes were active when the cell was alive. After Kim finished their game of electrical telephone, she carefully slurped out the nucleus from each of the 7 neurons. All the information gathered from these rigs can help researchers identify neurons that might play a special role in making a human mind. Such scrutiny, for instance, reveal what researchers think is a rare cell called a von Economo neuron, named the Austrian neurologist who first described the cell type in the 1920s. The extra long, extra spindly neurons was found in the live brain tissue donated by a 68-year-old woman who had surgery to remove a tumor. The neuron displayed an unusual electrical response to the current applied to it. The Allen Institute Science colleagues reported online May 7th at biorxiv.org. The results was tantalizing because problems with the von Economo neurons are suspected of playing a role in psychiatric conditions and Alzheimer's disease. Studies on live human. Okay. Anyway, studies on live human cells also turned up an important difference between humans and mice. A certain kind of human neuron is covered with a protein called an H channel. In mice, those channels are rare. H channels help cells respond to electrical signals and can be affected by drugs, including one for epilepsy. This basic difference between the 2018 in neuron and might explain why certain kind of drugs work differently in the brain of mice and people. More broadly, these newly discovered properties of human neurons might be the thing that enable some of the most sophisticated features of our brain. Taking stock of live human neurons is essential, not just to satisfy humans' navel-gazing curiosity, says Nadad Stestin, a Yale School of Medicine neuroscientist. Discovering the quirks of human brains might lead to us understanding one day why we suffer from certain disorders, Stestin says. In precise, animal models have stymied research of schizophrenia, autism, and Alzheimer's disease, he says. That's why studying live human tissue is so crucial. All right, from brain to the lab. Allen Institute scientists fly. Goodness, everything wants to pop up when I read. Allen Institute scientists fly into the action when a brain sample arrives from a nearby hospital, bringing as much information as they can out of the precious tissue. What the researchers learn about the cell's behavior, shape, and genetic activity is shared publicly in the Allen Brain's Atlas. Bold text shows the steps of duration. 1 to 3 minutes surgery. The brain tissue is removed at a Seattle hospital, usually within a minute of the surgery, opening the patient's skull and placed into a jar with chilled artificial cerebrospinal fluid bubbling with oxygen and carbon monoxide. 15 to 30 minutes transport. A specifically equipped van speeds the tissue from the hospital to the institute. All right, 30 to 60 minutes slice. The sample is placed on a dish in a bucket of ice, trimmed and sliced into anywhere from 6 to more than 30 flakes. All right, that are each 0.35 millimeters thick. Distribute. Each sample, one slice, goes into a freezer for future analysis. Damn. Oh, goodness gracious. Each fifth slice is fixed and preserved. The rest are kept alive for more same-day experiments. 1 hour rest. Live cells recover from the trauma of surgery. Observe behavior. Rested cells move to electrophysiology lab, where the rig operators study cells' behavior within 30 minutes of electrical prodding. Map cells' shapes, filling some neurons will die. Researchers can see the elaborate projections. Study of genes. Thin glass pipettes pull out of the nucleus of some neurons so researchers can see which genes were active just before the cells died. Weeks to months, manipulation. Other tests attempt to insert genetic material into the cell that stays alive longer than expected. The cells glow for better visibility. A blow to the ego. But human brains aren't always so unique. A new result might disappoint people who think that our brains are teeming with specialized neurons that let us talk and think in ways other animals can't. The overall number of cell types in the human cortex and in the mouse cortex is roughly the same, says the study led by Allen Institute researchers that is in Impressive Nature calls the finding the biggest result to my mind. People, including scientists, have this strong need for human exceptionalism, Koch says, but the fact that the overall resident population of the human brain, mouse brain, is remarkably similar, based on the brain tissue from surgeries as well as the post-mortem tissue, adds to the list of the blows to the human ego. First, Darwin downgraded humans to just another animal tree of life. Then the Human Genome Project shocked us with the news that we have a similar number of genes as mice and fewer than water fleas. That's crazy. Now add the brain cells to the type list of things that make people more like mammals. Unbelievable. A piece of the brain is taken from the surgery patient left, obtain what may be rare. These large spinally cells, right, show that there's different dyes might enable some human sophisticated abilities. All right, that's crazy right there. Y'all see that? The paper coming out in Nature is historic, says co-author Raphael Youssef, a neuroscientist at Columbia University. In terms of understanding how humans compare with other animals, it's going to be a before this and after this. Those similarities don't surpass Susanna Herculano Huzel, a neurobiologist at Vanderbilt University in Nashville. Oh, Tennessee. Anyway, we are not special, so she says. Finding that humans and mice have similar types of cells in the brain makes a lot of sense, as does the idea that some cell types and some genes will be species specific. That's tongue-tied. Some genes will be species specific. The question is, she says, which of those differences are actually meaningful, all right? The explanation for why we are so smart, then, not that our brains are teeming with specialized human-specific neurons. The answer must be found elsewhere. Perhaps it really is a small number of rare neurons, such as von Economo neurons, or in neurons that haven't yet been discovered. Still, other scientists think that our brain power might come in part from cells in the brain that are neurons, such as the glial cell that perform a range of basic brain jobs that scientists are just beginning to understand. So maybe, as you said, I suspect the answer in the sheer size of our brain compared with our relatively small body, or perhaps our smarts, are a result of our long lifespan and the fact that we are immersed in cultures rich with language, literature, customs as Herculano Huzel points out. Alright, to make it easier, we find rare cell types. Scientists infect live human brain slices with viruses engineered to cause a certain group of neurons to glow red and green under a microscope laser. That looks crazy. You see that? Mind fix, alright? As D did his slicing, neurobiologist Jonathan Ting waited eagerly for his bit of brain. Ting runs experiments that deliver gene to live cells, as he kept this cell alive a surprisingly long time. In his fourth floor lab, Ting is not happy with the piece he got, calling it kind of a bloody mess. but he cuts it up some more and returns the resulting slices to the bubbling solution. Then he pulls what looks like a baking tray out of a nearby incubator, makes a Martha Stewart joke and shows me some live brain samples that are a week old. Some have survived for several months, a hardness that shocked the researchers when they first began these experiments. This kind of durability comes in the hands of as teens tinker and with how best to infect of cells with viruses. His goal is to use the viruses to deliver the genes that make certain groups of live human cells glow. The glow makes it easier to study the cell and ultimately to figure out how to change their behavior. To Ting's delight, he discovered that the virus and its luminescent, I'm sorry how you say that, cargo can be delivered simply by dropping virus-laden liquid on the blood-brain slices, okay? As Ting and I looked at glowing red and green cells on his microscope monitor, he describes the potential of his work with viruses. Not only will the researchers be able to find rare cells into the human brain, but they might be able to ultimately control the cells too. Imagine if von Economo cells, for instance, could be turned on and off at will with methods that are already under development in animal models. All right. If the cells are actually involved in a disorder, such as schizophrenia, then this sort of precise control could lead to a targeted treatment that toggles the cell's activities up or down as needed. It could also ultimately reveal how information flows through these cells in a way to make up the mind. All this tinkering has already turned up big findings, but there is a vastly more uncharted territory to explore, Kai says. The brain is by far the most complex, highly organized piece of active matter anywhere in the universe, all right? So pretty much, if you want to read more about the comments and things that people had to say, this is a very, very, very detailed article. I ran across this, and I wanted to, share this information with you all. What do you think about what scientists are doing with the pieces of the brain and how they actually are, coming up with ideas to make use of them, if that makes sense. You know what I'm saying? And I think this is a very interesting thing. Who knows that they've probably been doing this for years, but let me know what y'all think. Peace.
Now that's Huggie Pacino certified.
Chicken and corn.
You want something to eat? I live for this shit. Why you used to have business, boy?